Okay, so welcome to this next video in which we are discussing the adaptive immune response. Okay, so we're now going to actually start discussing the adaptive immune response. We've just had a revision of the acute inflammatory response. Okay, right, so the adaptive immune response then. So the first thing to understand is that there are two major types of adaptive immune response. There is one known as the cell-mediated immune response. Okay, and there is another type of adaptive immune response known as the humoral immune response. So humoral immune response. Okay, so what do these mean? Well, in my opinion, these are confusing names. You could have named these simpler. The cell-mediated response, brutally, is the response to intracellular pathogens. Okay, whereas the humoral response is the response to extracellular pathogens. Now, what do I mean by that? Well, if we, for instance, look at viruses as a pathogen, viruses go inside the cell. So if we've got a normal human cell here, what's going to happen is that viruses are going to go into this cell. So let's say we've got our virus here. Basically, the virus is going to end up inside the cell. So this is the virus particle here. Okay, so basically, it's a more extreme infection because the cells of the body are actually going to be infected by the pathogen, whereas other pathogens, for instance, let's have some bacteria. Those will just live alongside the cells. They won't try and go inside them. Okay, and that bacteria is far too big. It should be tiny compared to the eukaryotic cell. Okay, I was trying to draw it relative to the virus, but of course, it's gone horribly wrong. Okay, so it should be tiny, and the virion particle should be even smaller, basically. So, this is a bacterium here, and an extracellular bacterium. Some bacteria are intracellular, and we'll discuss some of those later, such as Mycobacterium tuberculosis is probably the most famous example. Okay, but generally, bacteria will be extracellular, so they'll be living in the tissue fluid next to our cells. Now, even though that's a less severe infection than this, we still don't want them there. We don't want them within the human body. But I hope you can appreciate that these are two different types of infection. This one, what you've got is the bacteria living in the interstitial fluid around the cells. This one, you have actually got a pathogen that is living within your cells. So basically, this is where you understand the difference between what we really are and a single life, because we humans, we consist of 100 trillion cells, so really we are 100 trillion lives. Okay, we're almost like a city of cells. We're what's known as a meta-organism. We're built out of a huge number of little organisms that are all working together in like a city, basically. Okay, so basically, intracellular infections are where the actual little organisms that make us up, the cells, are actually infected themselves. They're actually ill now. Whereas extracellular infections is when there's just some invader within the city, basically. There's someone in the city that shouldn't be there. Okay, so these two different types of infections require different adaptive immune responses. And the response to extracellular pathogens is going to be the humoral adaptive immune response, whereas the response to intracellular pathogens is going to be the cell-mediated adaptive immune response. Okay, right. So, later on we are going to have to separate these two. However, for the initiation part, the cell-mediated and the humoral immune responses are almost identical, basically. So we're going to keep them going alongside one another for the time being, although later on we'll have to split apart. So later on we'll have to first discuss the humoral response, and then we'll discuss the cell-mediated response. Okay, right. So let's begin with how you initiate the adaptive immune system then. Okay, so it starts off with a professional antigen presenting cell. So let me explain what a professional antigen presenting cell is. So the main two types of professional antigen presenting cell are macrophages and dendritic cells. And I should have said those the other way around. The main type is the dendritic cell, and then to a lower extent there is macrophages. As we'll see later, B cells are also antigen presenting cells, professional antigen presenting cells. But we'll start off with the dendritic cells and the macrophages, because they're usually what are going to initiate the adaptive immune response. So, basically, all cells in the body can actually present antigens, which is the process where you chop up an antigen, okay, and you stick a fragment of that antigen on the surface of your cell. Now you might wonder, well, what is an antigen? Now an antigen is just anything which is capable of having an antibody made against it. Okay, so it's anything that's capable of triggering an adaptive immune response, basically. So they're usually proteins. So really what we're going to do is we're going to chop up proteins from the pathogen and stick fragments of them on our cell membrane, bound to another protein which we'll come across in a moment, known as the major histocompatibility complex. Now all cells can do this process of antigen presenting, but they're not capable of activating the adaptive immune system, and I will explain why in a moment. Okay, so let's begin the story then. So we have our dendritic cell or our macrophage. Really the dendritic cell, but we're going to use the macrophage simply because of the mundane reason that it's easier to draw a macrophage than a dendritic cell. Here, remember, is the structure of our dendritic cell, this structure that looks rather like a starfish, and it's just really awkward trying to show these interactions on a dendritic cell, which is why we're going to do it with a macrophage, a simple blob of a cell, rather than the dendritic cell. Okay, so here's our dendritic cell, and here's our other professional antigen presenting cell that potentially can initiate the adaptive immune system, although it's most likely going to be the dendritic cell. However, as I say, we will do all of our diagrams with the macrophage simply because it's a nice sensible shaped cell rather than the dendritic cell. Okay, so what's going to happen then? Well, basically, both of these cells are phagocytes, and dendritic cells are one of these more rare forms of phagocytes, okay? So the macrophages and the neutrophils are the main two phagocytes, but dendritic cells are also capable of phagocytosing pathogens. So, this is where we need to understand that there are two different types of infection. There is the cell-mediated intracellular infection, and there is the humoral extracellular infection. Okay, so basically let's begin with the extracellular infection, and we'll also make a comment about the intracellular. Okay, so if the bacterium is extracellular, then what you're going to need to do is you're going to need to phagocytose the bacterium. Okay, so we'll show our macrophage doing this. So this is for the intracellular case, and we'll draw the cell-mediated one in there. And by the way, these names do make sense because humoral refers to the fluids of the body, and that's because the pathogen is within the fluids of the body. And cell-mediated means that they're inside the cell. So these names do make sense after you realize what they're talking about, basically. Okay, so the macrophage is going to phagocytose our extracellular bacterium here. So, let's show that happening. So here is our macrophage, and basically it's phagocytosed the extracellular pathogen, whatever it may be, but of course you can think of a bacterium to keep this simple. Okay, so I'll move this up a little bit. Right, so this is the extracellular pathogen in the phagosome now. And this is the nucleus of our macrophage here. Right, now what's going to happen is this bacterium is going to be broken down, or this extracellular pathogen is going to be broken down, and many proteins that are associated with this bacterium And these proteins we will now refer to as antigens. So these are proteins which we are capable of launching an adaptive immune response against. We're capable of making antibodies against them. So there are certain proteins on the surface of the bacterium that we are going to say that protein looks nothing like a human cell protein. We could target that, basically. Okay. Now these proteins will be broken down in the process of the destruction of this microbe, okay, and we'll get loads of fragments of this antigen. Now, what we will do is present certain fragments of the antigen, so certain little pieces of the antigen, on a protein on our surface, okay, and it's actually a protein complex, but we won't worry about the details, okay, and this protein complex on the surface of the antigen presenting cell that you're going to load this antigen fragment onto is known as the major histocompatibility, and I left just the wrong amount of space to fit in histocompatibility there. Major histocompatibility, which is a massive great word, complex class 2, okay, and because major histocompatibility complex class 2 is a right mouthful, often this is just abbreviated to MHC class 2, okay, so for short, M is for major, H is for histocompatibility, C is for complex, and then we've got class 2 on the end there. Okay, and because it's such an important molecule, everyone always puts that 2 in Roman numerals. Now, basically, we are going to stick a little fragment of the antigen in this little space that I've shown here. So we're going to put a snippet of our antigen in here. So this is a fragment of the antigen. So the shorthand for antigen is to write AG for short. Okay, so this is an antigen fragment, okay, and it's now sitting in this little cleft within the major histocompatibility complex. So let me get my highlighters on this. So in vivid purple, this is this major histocompatibility complex class 2, okay, and in fact I might as well colour the whole thing in now, and then what colour can I do that would show up? I think I'll do it in blue now. The antigen fragment is in blue there in the middle. Right, okay, so this is the process that you undertake to present a fragment of an antigen of an extracellular pathogen, and I will stress that to express the fragment of an antigen of an extracellular pathogen, you had to phagocytose that extracellular pathogen first, so that you could then break it down. This is not going to be necessary in the intracellular case, because if you've got an intracellular pathogen, such as a virus, all you have to do is wait for the virus to infect you, basically. It will come into the cell of its own accord. So we'll discuss antigen presentation of antigen fragments for intracellular pathogens in the next video.